Good afternoon, everyone. Thank you for joining our webinar today. This will be the first of BioNordika HTG Molecular webinar series. This will be the first session about the HTG Transcriptome panel and applications. Feel free to post any questions in the questions box. We can get back to you afterwards. So we'll have our guest speakers, Selina Gertner, who is the Director of EU Medical Affairs at HTG Molecular Diagnostics, and Professor Hans-Ulrich Schildhaus, who is a pathologist at the Institute of Pathology, University Hospital of Essen. Feel free to post any questions you have and give us your feedback. My name is Miguel. I'm a product specialist at BioNordika Sweden, and I will be hosting together with Selina. So without further ado, I'm going to give the stage to my colleague Selina. Please do. Thank you, everyone, and welcome.
Thank you so much, Miguel, and thank you everyone for joining our webinar series today. I'm very excited to kickstart this new series we will be doing on a monthly basis with new exciting topics about applications on HTG at SIG. And today we're going to present to you the HTG Transcriptome panel and an exciting application with a presentation from Professor Hans-Ulrich Schildhaus on sarcoma. So I will start with a short introduction into HSeq and the transcriptome panel. We will then have Professor Schildhaus presenting his HTG transcriptome panel data that he revealed fusion type dependent biomarkers that are impacting overall survival of synovial sarcoma. He will then present live for Q&A session, and we will then close up on the closing remarks. So anyone also watching this recording later, feel free also to reach out to us individually as a follow-up of this session. But also for the attendees, you can use the questions box to ask any questions during the presentations. So let's start with the introduction. My name is Selina Gertner, Director of Medical Affairs at HTG Molecular Diagnostics, and I will be introducing the technology and the application of the transcriptome panel. So what HTG provides is everything around the HSeq system, and the HSeq system ultimately allows multiplexing molecular profiling with very little sample amount. And what comes with this system is any reagents and consumables to run it locally on site. But there's also laboratory service, which is called Verio, that supports the analysis for a service that you simply send in your samples for the analysis. And for both applications, there is a data analysis suite that is called Reveal available that facilitates the complex data analysis of gene expression data. And the power of precision medicine is really on the RNA level, and that's where HTG focuses on. There is great value in DNA, and DNA is a very valuable biomarker and has been around for a long time. And it's a great tool to analyze also mutations for precision medicine. And the challenges you might have is that on the DNA level, there are mutations that are not actionable. And therefore, there is limited value in actionable mutations that are not identifiable. With the RNA, you have the advantage that you really see what's happening, what's basically live happening in the cell that is transcribed. And that also brings the value of multiplexing with it. So you can look at many RNA transcripts at the same time, and therefore you can detect the mechanism of disease progression or development on the go, so to say, while the cell is transforming that into the messenger molecule, the RNA. Obviously, protein is the executor, so therefore that's the gold standard for disease detection. But the challenge with protein is the multiplexing, so you cannot easily look at many proteins at the same time. And that's where the RNA really bridges the values and combines the values of RNA and DNA. And that's where HTG positions itself as an expert in RNA analysis. The sample amounts are getting less and less, and the approaches are being less and less invasive. So our surgical resections are being more and more limited, and there's more and more the approach to go to needle biopsies, and also more focus on liquid biopsy sampling, and HTG can support all that along the line. So many different sample types and very little material that is required. Just an overview here about sample input amount. If I talk about low sample input, I do really mean it. So if we talk about FPE tissue, one single section of 5 micron thickness is sufficient, where we use 6 to 11 square millimeter, depending on the panel. And also liquid biopsy, you can see here sample input is really low. You have extracted RNA listed here, but we prefer to work extraction free. If you have extracted RNA, you can still use it. So why extraction free, and what is this all about? So the beauty of extraction free is that basically you avoid any loss of material and also biases that are associated with RNA extraction. So there are standard kits that do also purify fragments on a particular length. So FFPE extraction kits go above 70 nucleotides. Anything that is higher fragmented will not be detected. And a standard RNA extraction kit even above 200 nucleotides. So the chemistry of HTG allows to detect nucleotides down to 50 nucleotides, and therefore highly degraded samples can also be detected. And it can also be that you lose material. So the gold standard RNA extraction kit actually assume 20 to 30% loss. And if you consider all of that, it could be that your valuable biomarker that is really of interest to your research and to your diagnostic application does get lost while you're extracting. So obviously, if you can now detect your target sequence directly in the lysate of your sample, that's a huge advantage. So we're talking about transcriptome profiling. Obviously, the first thing that comes in your head is probably RNA-seq, because it's perceived as a gold standard for RNA profiling analysis. And therefore, we want to compare here how the workflows compare. And you can see on the first look already, the workflow is much shorter because, as I mentioned, the extraction is removed, so you avoid all these steps. And then the majority of the steps are automated, so that you have also less hands-on time and shorter turnaround time. You limit it then to about 3 days workflow, and compared to RNA-seq, you have to 4 to 8 days. Obviously, that then also reduces the labor and the cost that is associated, and it also allows you to have faster access to your data. So the chemistry, just really high level, here's the quantitative nuclease protection assay. How does it actually work is that we simply add a protection probe, highlighted here in orange, in the lysate. This protection probe hybridizes to the target sequence. And in the next step, S1 nuclease digests any unbound probe or single-stranded molecules, so that ultimately what comes off the HTG platform is a protection probe that hybridizes to your target sequence. And this sequence is then included in the library preparation, where you add sequencing barcodes and adapter, and you can then quantify and pool and sequence. So traditionally, we use Illumina or Thermo Fisher platform for sequencing here. So let me talk about the transcriptome panel. That product was introduced last year, and it's a huge improvement on what HTG provides, because we are now going to the almost whole transcriptome with up to 20,000 transcripts we are looking at the same time. So you have 20,000 target probes in there, and we're detecting therefore almost the whole transcriptome on the mRNA level. And that also brings with it the added value that I already talked about, like the low sample input amount, the high success rate on FFPE, so highly degraded samples, the simplified workflow and the fast turnaround time, and also the bioinformatics is much simpler with a targeted approach. So with this comparison study, we want to actually look a bit on how it performs. You will see in a bit the presentation from Professor Schildhaus on what he achieved. But we also really wanted to test how does it actually compare, for example, in terms of success rates, like sample pass rates, but also in terms of archival FFPE tissue. If you have like 10-year-old blocks, how is actually the performance. And you can see here transcriptome panel versus RNA-seq. First of all, there's the extraction-free approach for HSeq, and then the extraction for RNA-seq. And you can see there was quite a good dropout already for the 5-year-old samples, so about 15% dropout. And for the 10-year-old samples, even more. So only 63% of the samples did actually pass, where HTG was able to generate good quality data for all of these samples. So in terms of poor quality samples and archival FFPE tissue, we can say that HTG panel is performing better and generating better results and higher pass rates on these challenging sample types. And I mentioned turnaround time in 3 days with the transcriptome panel, and RNA-seq it took 7 days. We also then want to see if you actually look at differential expressions. So we have the log scale here on the X and Y axis on the log differential expression of RNA-seq compared to transcriptome panel, and basically simply correlate how well is the differential expression correlating on these two platforms. And you would expect it on the blue line, and you can see there's really good correlation with a Pearson correlation of 0.83. If you consider the different chemistry and different technologies, the accuracy or the precision here is quite good in terms of the correlation of a Pearson correlation of 0.83. Data analysis can be really challenging, and therefore we also have the Reveal software here available to analyze your data. You can perform QC analysis, correlation analysis, box plots on most differentially expressed genes, heat maps, volcano plots, PCA analysis, see how your samples are clustering. So it really is an easy to use tool where you don't need highly skilled bioinformatic experts that analyze your data, and you can get high quality publication quality figures out of this software for your analysis. So simply to also conclude here a bit on the presentation before we then move on, I would like to highlight that the transcriptome panel really redefines RNA-seq, because you're leveraging the increased sensitivity, the simplified workflow, and the cutting-edge performance on HSeq, and you're expanding it now to 20,000 transcripts, and therefore covering not only the transcriptome, but also you're able to even look at the miRNome in parallel. So I think there's a great advantage that HSeq now provides with this transcriptome panel. And with that, we will then move on to the presentation of Hans-Ulrich Schildhaus. He is at the Institute of Pathology in the University Hospital Essen in Germany and West German Cancer Center. And he's going to present to us the HTG Transcriptome Panel revealed fusion type dependent biomarkers impacting overall survival in synovial sarcoma. Please note this is a recording of a presentation Professor Schildhaus did at the UK Biomarkers Congress in end of 2021, but he will be joining us just after the recording for the Q&A session, so please be ready with questions as soon as he will be live then.
Thank you, Selena. Everyone, just give me please a little bit so I can share the presentation. Please let me know, give me your feedback in any way you can if you do not see or do not hear the video. Okay, I'm going to start sharing the video now. Thank you.
Thank you very much for this kind invitation to this meeting here. I'm Hans-Ulrich Schildhaus. I'm a pathologist and molecular pathologist from Essen, Germany, and from the West German Cancer Center. And as you will see, I have a specific focus on sarcoma pathology as well and molecular diagnostics of sarcomas. And therefore, I'm very happy to share some still preliminary insights in what we did with synovial sarcomas, a very interesting sarcoma subtype. And we have analyzed them with a novel and recently introduced transcriptome profiling approach. Here are my potential conflicts of interests. You can see personal things, institutional things, and I would like to stress that this study, which I'm going to present here, has been supported by HTG Molecular Diagnostics. So a little bit, a few words on synovial sarcomas and on the background of this. Synovial sarcoma is a group of soft tissue sarcomas which are usually clinical aggressive, and unfortunately it affects usually young patients, adolescents, young adults, the so-called AYA group of patients. So that means this makes this tumor a very interesting, very important field of research to improve diagnostics, to improve treatment options, etc. So beside this more general clinical relevance of this tumor, most synovial sarcomas are clinically aggressive in terms of local relapses, which can occur simultaneously or metacuriously, and or distant metastases. And interestingly, there are clinical different subgroups which have been known for years by experienced clinicians. Some of the patients suffer very early from a very early relapse or very early distant metastasis, but there are others where the progressions occur very lately, sometimes even after 20 years of initial diagnosis. This is another interesting clinical information, and on a molecular level, this tumor is a paradigm for gene fusion-driven cancer. These synovial sarcomas have 100% of these tumors have these SS18 gene fusions, and 100% of SS18 fused tumors are synovial sarcoma. So this makes this very clear in terms of the underlying biologic event. So there are, however, at least two different types of gene fusions occurring there. One is the SS18 fusion with SSX1, and there's another group which accounts for roughly 1/3 of the overall cases, which is where SSX2 occurs instead of in SSX1. Very rarely also SSX4 can occur as a translocation partner. So what do these chimeric fused proteins do in these tumors? And there has been substantially increase of our understanding of these tumors. We know that these SSX, SS18 gene fused or chimeric proteins interact with a SY-SNP, the SY-SNP complex, and these interact with this KDM2B, which is an histone demethylase. And this is part of the component of a gene repressive complex, the so-called PRC1.1 complex. And this whole interaction leads to an aberrant activation and aberrant expression activation of genes which are developmentally regulated and which are usually repressed in adult tissues. And this recapitulates in immature, more embryonic-like type of gene expression levels. So that means we can say this SS18, SSX1 fused or chimeric protein deregulates developmental programs by hijacking of the transcriptional repressive complex, the PRC1.1 complex. So this makes this a good candidate to be studied by gene expression analysis. So since we would expect that these synovial sarcomas would express a completely, more or less completely different repertoire of genes compared with other sarcomas, which are not driven by these specific SS18, SSX1 or 2 gene fusion. So these genes or these fused gene consists of a major part of this SSX or SS18 gene, which has been formerly known as SYT. This has been renamed as to SS18 since this is located as on chromosome 18. So that means the major part of this chimeric protein consists of this SS18 component, and there's only a smaller component which is contributed by either SSX1 or SSX2. And interestingly, both proteins SSX1 and 2 are both different chimeric. The chimeric proteins differ in only 13 amino acids. So that means there's a really tiny difference between those different versions of this gene fusion. So I think this makes it plausible that both translocation subtypes lead to the same tumor subtypes. However, it has been demonstrated by several groups that there are obviously, from a clinical perspective, also some differences between SSX1 and SSX2 fused synovial sarcomas. For example, the SSX2 rearranged tumors do show a little bit more predominance of female patients. On the other hand, these biphasic morphology, which is defined by a spindle cell and an epithelioid component, is underrepresented in the group of SSX2 tumors. Also in terms of the overall survival, both groups seem to differ a little bit. So as you can see here in these overall survival curves here, and this should or is obviously independent of the occurrence or non-occurrence of metastasis at the initial diagnosis. So, however, these findings have not been confirmed by other groups. However, our clinical experience and our clinical experience points toward that among those SSX2 patients, there's the higher likelihood to have these late responses and to have to survive longer. So that means this makes it interesting for us, and we wanted to define novel biomarkers which allow us to further study these differences between these tumor subtypes. As already mentioned, there's this change in this gene expression repression makes this tumor interesting in terms of gene expression analysis, and this has been demonstrated already or has been studied already in previous approaches. You can see here one example of studies which have been published based on cDNA microarrays, demonstrating that the group could demonstrating a nice difference between MFH, this is malignant fibrous histiocytoma, which stands for an undifferentiated pleomorphic sarcoma, and synovial sarcomas. I think this is not so much surprising. They also demonstrated some of those genes which have already been demonstrated by others, for example, AXL. And they could demonstrate also nice difference between biphasic and monophasic tumors, and this is also not so much surprising, since this is mainly driven by keratins. And these keratins are those proteins which make these epithelioid morphology in these tumors. However, the authors state they could not demonstrate any difference between SSX1 and SSX2 gene fusions, and they state here this negative finding may be unexpected given the known correlation of SYT fusion subtype with survival and areas of glandular epithelial differentiation, but the study group and the technical platform may have been inadequate to detect the likely much smaller difference in gene expression. So this makes it interesting for novel developments, for also in novel technologies. So, so what we aim to do here is we wanted to explore how comprehensive gene expression analysis can help to define synovial sarcoma subgroups, and we wanted to determine how synovial sarcomas as a group differ from other sarcomas based on in entire picture, comprehensive picture, and we have chosen two different comparison groups. One is in sarcomas with other types of gene fusions, what we call fast sarcomas here in this study, and we wanted to compare these with the morphologic mimickers, other spindle cell sarcomas without any known genetic alterations. And we wanted also to identify differentially expressed genes between SSX1 and SSX2 subtypes of synovial sarcomas. What did we do? We did a retrospective analysis of archival material. The paraffin blocks were up to more than 4 years old. They have been stored under standard conditions at room temperature. No specific tissue. We have used resection specimens and biopsy specimens, with the majority of samples representing resection specimens. We have included 45 synovial sarcoma samples with known fusion status, and the comparison sarcomas 23 spindle cell sarcomas, NOS, and 8 fusion positive sarcomas other than SS18. So the technology what we have used was the HTG Transcriptome panel, which is more or less based on this nuclease protection assay procedure methodology, which is basically a method to determine, just to count copies of RNA in tissue without any extraction and without larger amplification steps. So basically, the target RNA is covered by probes which are containing these wings and wingman structures, and this and the entire lysate is digested by the S1 nuclease, which digested anything of nucleic acids but keeps those DNA RNA chimeric structures there, and this is how they are kept there in these lysates, and then you can easily adapt ligate adapters there, and can, with a few amplification steps, you can just count the amount of these numerically these molecules. This panel, what we have chosen, contains roughly 20,000 probes, including controls, and we used 1 or 2 sections per block, only 1 section. So this is a nearly fully automatized workflow. The majority of things are done on this HSEC device here. And then we have finally used an Illumina NextSeq 550 sequencer for reading on counting the number of molecules there. This platform has some advantages over RNA-seq, especially this simplified workflow. This is really something what we appreciate with this technology, and has a high pass rate, as I will demonstrate. And this is higher than on RNA-seq from FFPE material. This panel comes also with some QC metrics, which we have also used in our cohort, which measure the RNA quality, the read depth quality, the background signal quality, and the DNA suggestion quality. And this is what we have seen here in our. From our cohort, we can demonstrate that based on the RNA quality, we can see that we have in all of those 76 samples, we have all samples in place. There was no issue with the quality. Here, in terms of the read depth, also all tissues met the criteria of quality, and also in terms of the background signal, we could accept all of those. So this was very good to see that also these archival materials contain a lot of high quality or reveal a high quality of samples. There were only 2 samples which we excluded based on incomplete DNA digestion. But these were only 2 out of these 76 samples. Based on a pre-specified probe set, we could demonstrate that we could nicely separate these different groups of sarcomas from each other. We could nicely demonstrate that the synovial sarcomas cluster quite close to each other. However, this wasn't, from my perspective, a moderate separation. These pre-specified probes that included also SS18, for example, other genes which have been published in the literature. If you look at the entire picture and use more than 10,000 genes simultaneously in this principal component analysis, we can nicely see that we can separate the spindle cell sarcomas from other fusion-positive sarcomas and from synovial sarcomas. Interestingly, there are some cases which are quite more similar to each other, and these are currently interesting cases which we are currently working on. And there was one outlier case which has been demonstrated a completely different gene expression profile based on a neoadjuvant treatment, which induced a lot of cell damage here as well. If we compare the spindle cell sarcomas versus the synovial sarcomas, we can exactly demonstrate what we expected, that there's a huge number of deregulated, differentially expressed genes there. More than 5,000 genes are differentially expressed, and this is exactly what we expected. And we can also look down to these different types of genes, and you can see as those what we expect. TLE1, for example, is dysregulated, etc., etc. So that means you can nicely identify also this gene of interest there. And if you go with the top 50 deregulated genes back to this principal component analysis, you can see close to perfect separation between these different tumor entities there. And this is a list of those genes which are deregulated between these genes. And again, TLE1, this is what we also use as a diagnostic marker, comes up here very well. But there are a lot of interesting genes what we are currently evaluating and validating with other methods. Also, in terms of SSX1 versus SSX2, we could make find an interesting observation here. And yes, we did find differentially expressed genes between those groups, SSX1 versus SSX2. This is only 13 out of those more than 10,000 genes which have been investigated there. And this is very interesting to see, and this is also something which provides us new insights into novel biomarker assays and biomarker development to separate these groups. Among them, there's also histone deacetylase, as you can see here. So that means this tool enables us to identify those few genes which are making the difference between SSX1 and SSX2. So with this, I would like to sum up here. Synovial sarcomas are aggressive spindle cell sarcomas showing this consistent translocation either with SSX1 or SSX2. And the overall survival is thought to be better in patients with SSX2 than in SSX1. So there is a need to further evaluate these changes since this is not yet so much clear clinically. And we are in these broad extraction-free transcriptome profiling help to identify differentially expressed genes that are linked to the translocation type, which provide us a novel basis for developing biomarkers for these subgroups. These differential expression profiles between synovial sarcomas and other spindle cell sarcoma types were in line with the previous findings. and the identified biomarkers offer new opportunities for improved diagnostics for aggressive synovial sarcomas. With this, I would like to thank the people who were involved in this project. I would like to thank my team from Essen. I would also like to thank the HTG team, which supported us a lot with all these technical things. Thank you for attention, and thank you for listening. And yeah, I'm open for any questions if there are some.
And at this point, hello, I would like to thank Professor Schildhaus for making this presentation available for us. It was a really great and interesting presentation. Thank you once more. So, Sabine, if you'd like to join me, we will begin a Q&A session now. So any questions you have for either Selina or Professor Schildhaus, please let us know. I can see that Professor Schildhaus is already here. Yes, if you can hear us. Seems he's connected to audio, so we should have sound.
Yeah, I can see he is joining, so probably just needs to deactivate the microphone. But in the meantime, yeah, maybe we can see what questions did come in.
So now I just find the button, the right one.
Perfect.
So
welcome.
I'm there.
Thank you so much for joining, and thank you again for this great presentation.
I actually have some questions. I have one question, which is for Professor Schildhaus. How do you plan to validate the fusion type specific gene signature?
Yeah, this is a very good question. Thank you for this. So there are multiple options to validate these things. One is basically to detect protein expression levels in the tumor tissue directly. Some of the things are already clinically applied. So I mentioned previously that one of those differentially expressed genes is TLE1, which is already used as a diagnostic biomarker, and this is something which is very easily to be detected also on the basis of immunohistochemistry. So in terms of the huge bunch of these more than 5,000 differentially expressed genes between these fusion mimickers and the synovial sarcomas, we are currently trying to understand which pathways are involved in this, and this is also something which is currently underway, and this is something where we are very much appreciating the options which are offered by this technology to let us have a look on the entire picture of these differentially expressed genes. And yes, the next step will be to analyze the pathways and then to analyze signaling nodes and of pathways, etc., on the protein basis.
Thank you very much for the comprehensive reply. I have another question, which is about what's your experience on the performance of very small FFP biopsy sample analysis?
Could you please repeat the question? I have some issues here with the connection.
This is the analysis of a very small FFPE biopsy sample. What's your experience in the performance of this sample?
Yeah, thank you. This is another very good question. Since we are sometimes we as pathologists and molecular pathologists are currently very frequently limited by the size of the available tissue. This is really an issue in clinically applied sciences, life sciences. And therefore, so we found out that also, yeah, smaller biopsies in the range of a few millimeters are enough for being analyzed safely. And yeah, this is a great advantage also of this technology. So this is it's a very important thing. We very frequently use these small biopsies, especially if you think of other tumor entities, for example, lung cancer, for example, etc., where it's very difficult clinically to obtain these small biopsies, and this is a very valuable tissue. And it's good to know that this technology also works with smaller biopsies.
I will ask anyone if you have questions, please write them now. But we have one more question, and then if it comes any other question during this time. This one is actually about the potential or the implications for therapy. You actually said that the synovial sarcoma had a quite unique set of differentially expressed genes that you probably weren't expecting to see regarding other sarcomas, and especially what you said in the end, that SSX2 has better survival perspectives than SSX1. So do you think that this will have different implications in terms of therapeutic targets?
Mm-hmm. Yes, so let me start with mentioning that I regard these differentially gene expression levels under or as two different types of biomarkers. One is the prognostic biomarkers, and another is predictive biomarkers. And cancer treatment of sarcomas is currently on the way to being more or to coming more related to what we call precision oncology. That means to apply targeted therapies, which would require more comprehensive understanding of the underlying genetics of these tumors, or also including gene expression levels, etc. So honestly, we are not yet there. There are first very interesting options, also at the level of clinical trials, including immuno-oncology studies, which are using CAR T cells, for example, based on the differentially expressed genes which are related to these tumors. One example are those cancer testis related antigens, for example, NY-ESO, etc. So that means comprehensive understanding of the gene expression levels of these tumor may also fuel those clinical trials which are using CAR T cells and other technologies of immuno-oncology. And the second point is the more prognostic parameter, and this study was more related to this one. So we try to understand which gene expressions which may more closely related to good or bad prognosis of patients. And this is also clinically meaningful since all these patient care after surgery is heavily influenced by these prognostic evaluations. And this is something what we are currently also evaluating by validating these differentially expressed genes, and we are currently trying to understand better which of those genes is, or which set of genes is the signature which predicts a good or worse prognosis in those patients. So that means, in summary of this answer, these gene expression profiles have both meanings. First, in terms of prognosis, and second, as a predictive biomarkers for tailored therapies of the patients.
Thank you very much. I have no further questions from the audience. Selina, I don't know if you have any questions from your part.
No, I think it's really beautiful to see, and thank you so much, Professor Schildhaus, for illustrating how you're using that data set also to drive biomarker discoveries that have prognostic and therapeutic impact to be validated in clinical trials and further investigated, of course. But I think that's where it starts, or basically where it leads to validation. So really beautiful piece of work, and happy to see how we can support you in this initiative.
Thank you.
Thank you very much for sharing that with us today. It's extremely valuable. So now I would like to give the presentation power to Selina once more, so she can share one final remark with everyone, which is...
Yes, so we mentioned this will be a series, so this was the very first event that we started. Just want to share full screen here that you can see the slides. So really appreciate you are joining for this webinar, and appreciate Professor Schildhaus for presenting the HTG Transcriptome panel and what he revealed there in fusion type dependent biomarkers for specific for synovial sarcoma. And we want to change gears for the International Women's Day on March 8th and introduce to you MPD, the breast cancer recurrence risk assay that can also be used for breast cancer subtyping. So stay tuned on this event. If you're interested in joining on March 8th, save the date and you can already register. So maybe one of the team members can copy the link in the chat so that you can directly register and make sure that you have that in your calendar if you're interested. And yeah, with that, I really would like to thank you for joining, and hope you enjoyed this. If you have any additional questions coming up, feel free to reach out, and looking forward to see you March 8th.
Thank you very much, everyone, for joining. A big thank you once more to Selina and Professor Schildhaus for being here today and for sharing their knowledge and presentations with us. And don't forget our special webinar on International Women's Day. We will be promoting it further, so we will be receiving emails with registration and the poster as well. But until then, if you have any questions, please feel free to reach out to me or to any of our BioNordika, your BioNordika representatives in the Nordics or city. Yeah. Thank you very much for joining today.
Thank you. Bye-bye.
Thank you.
Thank you very much.
Bye.
Have a nice day. Thank you. Bye-bye.
Thank you. Bye-bye.
Bye.